Hello everyone, I'm Mr. Furlong, and today we're talking chromosomal inheritance. This idea of chromosomal inheritance actually generates genetic variations through three different processes: segregation, independent assortment, and fertilization. We've already talked about the first two. Remember that the law of segregation states that when chromosomes are separated during meiosis, the alleles will get separated as well. So the offspring is only going to get one of those two alleles, which one they get is totally by chance. Independent assortment says that two or more alleles separate independently of one another. So these two alleles for a particular trait, they're not going to stick together, usually because they are found on different chromosomes. The third one, though, is fertilization. Now, what fertilization is saying is that the sperm is going to fertilize the egg. Which sperm actually fertilizes the egg and which egg was released is totally random. So this can generate a large amount of genetic variations even if you have the same two parents. But we're looking at chromosomal inheritance. And the basis of inheritance provides an understanding of the pattern of the transmission of genes from parents to offspring. So here I have two different sets of chromosomes. This is from a human, 46 chromosomes. I made the mom's chromosomes pink and dad's are blue. They're not really that color. And by chance, mom is going to donate half of her chromosomes through meiosis. So she'll be donating 23 chromosomes. Dad will donate 23 chromosomes as well. And the offspring will have the required 46 chromosomes. So whichever genes are found on those chromosomes, that's going to be the genes that they get. So we've been looking at doing Punnett squares in the past and looking at specific alleles. However, certain human genetic disorders can be attributed to the inheritance of a single affected or mutated allele or specific chromosomal changes, such as non-disjunction. In the past videos, we've been looking at specific alleles. Well, this is going to be an allele on a chromosome. And chromosomes don't always separate like they're supposed to, and that can cause some issues. When chromosomes do not separate like they're supposed to, that is called non-disjunction. That is the failure of homologous chromosomes to separate during meiosis. Let's take a look at an example of this. Remember that we're going to donate 23 chromosomes. I'm just going to show two chromosomes right now just to keep this simple. The X and the Y. So let's say that this is a male. Now remember, at the beginning of meiosis, these chromosomes will replicate. And we should get, at the end of meiosis 1, two sex chromosomes in each gamete. Now they will undergo meiosis 2, and what's supposed to happen is that they separate like they're supposed to. These, of course, are going to become sperm cells. And so this would have 23 chromosomes, has the X chromosome. This one has 23 chromosomes, would have the Y. But during non-disjunction, they don't separate. So they stay together. So that becomes a sperm cell that has now 24 chromosomes. And this sperm cell only has 22 chromosomes. In the case of humans, whenever we don't have 46 chromosomes, that always causes some issues. In most cases, it is fatal. The developing fetus simply won't survive, but there are some exceptions to that. We're going to take a look at just a couple of them. To check to see if non-disjunction has occurred, you can actually have a karyotype done. This is where the doctors will take some amniotic fluid, they'll remove some of the fetal cells, grow them in a petri dish, and then when they undergo mitosis, when we can see the chromosomes, we'll take a picture of those chromosomes. Now, the picture, of course, isn't these chromosomes all lined up nice and neat. We do that ourselves digitally. And we simply look to see, are there two chromosomes at every number? And if you take a look, for instance, number 1 here, there's two chromosomes. There's 2 at number 2. So I look to see, and there are 2 chromosomes at every number. However, there are 3 sex chromosomes. So this individual has 47 chromosomes, and they are XXY. So then the question is, are they male or female? Well, they're actually male, because they have a Y chromosome. There is a gene on the Y chromosome called the SRY gene that determines the sex of the baby. This is known as Klinefelter syndrome. If we look at another example, we can see that there are 2 chromosomes at every number, but in this case there's only 1 sex chromosome. This means that this person has 45 chromosomes, not the 46. This is the only case where a person can have 45 chromosomes and still survive. They would be female because they don't have the Y chromosome, and this is known as Turner syndrome. Another common example of non-disjunction is, again, we can look to see that there are 2 chromosomes at every number except at number 21. So this person would have 47 chromosomes. They would, in this case, would be a male, and they have that extra chromosome at number 21. This is known as Down syndrome. Typically, having an extra chromosome at number 21, person does survive. If this were to happen at any other numbered chromosome, they almost never survive. So let's take a look at an example of using a Punnett square when non-disjunction takes place. Now, these are all going to be sex-linked traits, and we're going to look at calico cats. In calico cats, black fur is codominant with orange fur. Both of these alleles are found on the X chromosome. Since they're both dominant, they're both going to be expressed. So in a calico cat, you have both orange and black fur. So if we were to do this Punnett square, we would take one parent, the calico female, and we're going to cross this, in this case, with a black male. And let's complete this Punnett square. So notice the possible offspring. You could have a black female cat, a black male cat, a calico female cat, or an orange male cat. But what if non-disjunction occurred? And now one of the sperm cells has both the X and the Y. If we do this Punnett square, what we get is a black male cat, a black female cat, but notice here, this would be a calico male cat. Almost all calico cats are female because they have to have 2 X chromosomes. But in the case of non-disjunction, you can have 3 sex chromosomes and have a calico male. Calico males are very rare and very expensive. So if you happen to find one, keep it. So those are just a few examples of chromosomal inheritance, and I'll see you in class.